Good afternoon all. So today we are going to discuss about electron transport chain in bacteria. So in the last class we have discussed about the Krebs cycle. So today we are going to discuss electron transport chain. So what do you mean by electron transport chain? It is a series of complexes that transfer electrons from electron donors to electron acceptors. And this transfer can be permitted through a redox reaction. And this redox reaction involves both reduction and oxidation occurring simultaneously. And this couples this electron transfer with the transfer of protons across a membrane, and the protons are nothing but H plus ions. So this protons are transferred from the electron donors to an electron acceptor, and this process is called electron transport chain. The electron transport chain, it is built up of different types of molecules such as peptides and enzymes. Then the electron transport chains of bacteria, and they are operated in plasma membrane, because normally it is operated in mitochondria in microorganisms, but in prokaryotes this mitochondria organelle are absent, so they may operate in plasma membrane. Some bacterial electron transport chains resemble mitochondrial electron transport chain. Some resemblance are present, but mostly they are quite different from eukaryotic electron transport chain. Example, Paracoccus denitrificans. It is a gram-negative facultative anaerobic soil bacteria, and in this the electron transport chain takes place. The flow of electrons through the electron transport chain, it is an exergonic process, which means energy liberating process. The energy from the redox reaction, what happens? It creates an electrochemical proton gradient, and this electron-chemical proton gradient, it drives the synthesis of ATP. So the energy from this redox reaction creating a gradient, and this may drive the synthesis of ATP. In aerobic respiration, what happens? The flow of electrons terminates with molecular oxygen being the final electron acceptor. So, in aerobic respiration, oxygen is the final electron acceptor. In the case of anaerobic respiration, the other electron acceptors are used, such as sulfate as well as nitrate also. In the electron transport chain, this redox reaction, we have told that redox reaction which may permits the movement of this H plus ions from the electron donor to electron acceptor, and this redox reactions are driven by Gibbs free energy state of the components. I think the Gibbs free energy you have studied in plus 2. So this Gibbs free energy, it is related to a quantity called redox potential. And this the complexes in the electron transport chain use or harvest the energy of the redox reaction that occur when transferring the electrons from the low redox potential to high redox potential. So this may create an electrochemical gradient. So this is how the Gibbs free energy are involved. That means in the redox reaction, the transfer of electrons occurs from the low redox potential to high redox potential. And this electrochemical gradient created that drives the synthesis of ATP. How? Through the coupling with oxidative phosphorylation with ATP synthase. The electron transport chain and the site of oxidative phosphorylation. So these are found on the inner mitochondrial membrane in the case of eukaryotes. The energy stored from the process of respiration in reduced components such as NADH and FADH, it is used by the electron transport. So the energy stored in the process of respiration, for example Krebs cycle, and this energy is stored in the form of NADH and FADH. And these are utilized by the electron transport chain to pump the protons to the inner membrane space, and it generates an electrochemical gradient over this inner mitochondrial membrane. In photosynthetic eukaryotes, electron transport chain is found on the thylakoid membrane. Here, light energy drives the reduction of components of the electron transport chain and therefore cause subsequent synthesis of ATP. But in bacteria, the electron transport chain can vary over species, but it always constitutes a set of redox reactions, and those are coupled with ATP synthesis through the generation of electrochemical gradient. We have already discussed this point, and generation of electrochemical gradient and oxidative phosphorylation through ATP synthase. The bacterial electron transport chain vary in electron carriers. So, example, in their cytochromes, so their number can be varied, electron carrier number, and are usually extensively they are branched. Electrons often enter at several points and leave through several terminal oxidases. So, they might leave through several terminal oxidases. Electron transport chain, and they are usually shorter, which takes place in bacteria, are usually shorter and possess low phosphorus to oxygen ratio, that is P/O ratio, than mitochondrial transport chain. The bacterial and mitochondrial electron transport chain differ in details of construction, even though they operate employing the same fundamental principle, but their mode of construction is quite different. So this is the electron transport chain in eukaryotes. An outline, that is cytochromes. These are the different types of complexes, cytochromes, etc. So we will discuss, and these are taking place in plasma membrane. So bacterial cytoplasm, etc. So the bacterial electron transport chains in eukaryotes, NADH is important electron donor, and the associated electron transport chain is NADH to complex 1 to Q, then to Q means quinone, then to complex 3, then to cytochrome C, then complex 4, then finally oxygen, where complex 1, 3 and 4 are proton pumps, while Q and cytochrome C are mobile electron carriers, and the electron acceptor is molecular oxygen. In the case of prokaryotes, the situation is more complicated because there are several electron donors and several electron acceptors. Donor dehydrogenase, then the another donor quinone, which gives oxidase, then acceptor, another donor that is cytochrome to oxidase reductase, that is enzyme reductase, and finally to acceptor. Electrons can enter the chain at 3 different levels. At the level of dehydrogenase, so this may enter the 3 levels at the level of dehydrogenase, and also at the level of quinone pool, and then finally at the levels of mobile cytochrome electron carriers through which electrons can enter the electron transport chain. These levels correspond to successively more positive redox potentials. So they may be responsible for more positive redox potential or to successively decreased potential differences relative to the terminal electron acceptor. In other words, they correspond to successively smaller Gibbs free energy changes for the overall redox reaction, that is from donor to acceptor. Individual bacteria use multiple electron transport chains, often simultaneously. So, there are certain bacteria using multiple electron transport chain, and also there are bacteria can use a number of electron donors, as well as number of different dehydrogenases and a number of different oxidases and reductases, and a number of different electron acceptors, not only the oxygen, in the case of eukaryotes. For example, E. coli, when growing aerobically using glucose as an energy source, uses two different NADH, that means NADH dehydrogenases, and two different quinone oxidase for a total of 4 different electron transport chains operating simultaneously. The common feature for all the electron transport chain is the presence of proton pump, and this proton pump is to create an electrochemical gradient over a membrane. The bacterial electron transport chain may contain as many as 3 proton pumps like mitochondria, or they may contain only 1 or 2. They always contain at least 1 proton pump itself. So, these are all about different types of proton pump I have discussed. Now next I am going to discuss about electron donors. That I will discuss in the next class. Thank you.